Okay, so is my screen visible to everybody?
Yes, ma'am.
Okay,
Yes, ma'am.
okay, fine. Um, so we'll start today's session. So we'll just a minute. Students are entering now. Fine. So today in today's class we will be discussing about the topics that we have studied in week 5, which is recombinant DNA technology part 1. So in the lecture 1 we have studied about DNA delivery in host, where we have studied about the concept of transformation, how the transformation was discovered, what is the mechanism of transformation, what are competent cells, how to prepare competent cells, how transformation was carried out, how to calculate transformation efficiency. Then apart from transformation, what we can do electroporation. Then what are the factors that affects the transformation efficiency and transformation in yeast. So these were the topics that were covered in the lecture first. So as we always do in our class, first we'll do the questions, and simultaneously with the questions we'll see the concepts which are related to the questions, and in the last we'll see some of the topics that you students could not understand in the previous classes lectures. So moving on, so the our first question is, dash bacterial strain was used by Frederick for the discovery of transformation. What is the answer? Which bacterial strain was used by Frederick Griffith for his experiment?
Streptococcus pneumonia.
Streptococcus pneumonia is the correct answer.
Pneumonia.
Okay. Now the next question is, what are the storage conditions for competent cells? 15 to 20% glycerol at room temperature, 30% glycerol at minus 20 degrees Celsius, 15 to 20% glycerol at minus 80 degrees Celsius, or 30% glycerols at 4 degrees. So what is the storage condition for competent cells? Tell me. Competent cells
Option
storage
D, ma'am.
conditions. Hmm?
Option D, ma'am. 30% glycerol at 4 degrees Celsius.
30% glycerol at 4 degrees Celsius. No, it's not the right answer. Think more. So competent cells actually, you know, so suppose you take a vial of competent cells and you thaw it. So whenever you are thawing competent cells, you have to always make sure that you thaw at 4 degrees or at on ice. So when you are thawing at 4 degrees, that obviously means that it is not stored at 4 degrees, first of all. Second thing is that once you are thawing the vial, after using, suppose if some of the cells are remaining, some of the competent cell is remaining, you cannot refreeze and rethaw it. So once you thaw one vial, even if some competent cells are left, you have to discard it, because you cannot thaw a competent cell vial more than once, because then the efficiency of transformation would be highly reduced. Okay, so competent cells, like, they are very much prone of getting this thing, their efficiency can be altered if you are rigorously pipetting also your competent cells. So whenever you are thawing competent cells, you put it on ice, and once it is thawed, immediately you add your plasmid DNA to it, and immediately you kept on ice only. So do not take it from ice up till the 30 minutes incubation is complete. And even if something is left, some cells are left, you just discard it. You cannot just refreeze and rethaw it. Okay. Anyways, so what would be the answer? What are the storage conditions for competent cells? So 4 degrees cannot be, because we thaw it at 4 degrees. So 4 degrees not the answer. Then anybody else who wants to give a try?
Ma'am, B.
No. The correct answer is C. 15 to 20 degree percent of glycerol at minus 80 degrees. So minus 80 always stored competent cells at minus 80 degree. Room temperature in 4 degrees is not even an option. Room temperature so is never an option. And minus 20 not, because suppose, so whenever you prepare competent cells, so you prepare competent cells in stock and then you aliquot it in many vials. Okay. So suppose you have made let's say you have made 30 or 40 vials of competent cells, so obviously you will be using it for a very long time. So for long term storage, obviously it is always beneficial to store at minus 80 degrees, and competent cells anyways have to be stored at minus 80 degrees only. Next question is, what is the purpose of giving heat shock during transformation? A. It causes stress in the competent cells through which transient pores are formed. B. Heat shock creates stress and causes the upregulation of factors which are responsible for DNA recombination and repair. C. It lyses the cell wall. And D. Relax the cell wall, which causes the upregulation of factors responsible for DNA recombination and repair. So,
Ma'am, option B.
no. Option B is
Then
Ma'am,
option
A,
A.
ma'am. A, ma'am.
Then option
A,
A.
ma'am.
no. Both the options are incorrect. The correct answer is option D. So that's why I give like this. So you have to understand, first of all, there are no transient pores that are formed. This transient pore formation is the concept of electroporation, where you are giving voltage. So voltage is making your plasma membrane permeable, and your there is a formation of transient pores. So transient pore formation is not happening, but the important thing which you need to remember during heat shock treatment is that it kind of it causes the swelling of the this thing cell wall. So basically, it relaxes the cell wall, and the factors which are responsible for the DNA recombination and repair are upregulated. So the correct answer for third question is option number D. And if you have any confusion, you can go back to the lecture, first lecture. There you will find also. So next question is, transformation efficiency is more when identify the best possible option. Covalently closed circular DNA of log phase, supercoiled DNA of lag phase, covalently closed circular DNA with calcium chloride method, and supercoiled DNA of log phase. So which is the correct answer?
Ma'am, option D.
Yes, option D is the correct answer. Can you explain also?
Ma'am, mobility of the supercoiled DNA is highest, and in log phase, the cell can, um, I mean, its ability to take up DNA is the best in log phase.
So basically, supercoiled DNA, as he said, what's your name?
Ma'am, Karthik.
Okay. So as Karthik said that in supercoiled DNA mobility is highest. Log phase. So what happens in the log phase is that we have studied that bacteria follows certain phases. So lag phase, log phase, stationary phase, and decline phase. So the maximum growth is experienced by the bacteria in the log phase. So lag phase is the time of adaptation. So whenever you are culturing, you are putting them in a new media, in new growth environmental conditions. So they will take some time to adapt to the environment. So that is your lag phase. So after they are adapted with the environment, now they will grow exponentially. That means their growth rate is greater than death rate. Okay. So in log phase, your growth rate is greater than your death rate. Then comes the stationary phase. So what happens that it's basically a graph of a batch culture where you are not continuously supplying the nutrients. So when you are supplying nutrients, there will be a time at which the nutrients will get exhausted, and then there will be no more nutrients left. But because bacteria are still very much metabolically active, so they will grow. But because nutrients are also not there, nutrients are also depleting, so they will die also. So here growth rate is equal to the death rate in stationary phase. And then comes your decline phase, where death rate is greater than the growth rate, because now the whatever culture is there, whatever bacteria you have they have completely utilized all the nutrients, all the growth factors, and nothing is left. Now they will start declining. So from the graph you can say that the maximum, so the best phase of bacteria where they are growing at their best and they are maximally metabolically active is your log phase. So therefore, when the bacteria are at best, then the transformation of efficiency obviously would be best. So your transformation efficiency is best when bacteria are at their log phase and they are super coiled. Next comes the general procedure of transformation. So I've just kept it for a revision purpose, because transformation, up till now we have studied about digestion. So suppose this is your insert and this is your vector. Okay, so now we will digest it. So for digestion we will use the same restriction enzyme. So if I am cutting my vector, if first of all I will amplify this particular gene of interest from my genomic DNA. So I have the whole DNA, whole genomic DNA. Okay, from this I want to clone this particular fragment. So I will amplify this particular fragment with the help of PCR. Now that I have amplified, I have my insert and I have my vector. Now what I have to do, I have to put this particular fragment of insert in this vector. So for that, I have to cut the vector insert also, and I have to cut the vector also with the same restriction enzyme. So first we did the digestion, where we have cut our vector and the insert with the same restriction enzymes. After digestion, this is your vector. There are some, and this is your insert and this is your vector. Now what we will do is, we will ligate it. Okay, so ligation. And after ligation, we will do transformation. So genetic engineering basic protocol is, सबसे पहले PCR amplification of the particular piece of insert. After your insert is there, you will do digestion, then you will do ligation, then you will do transformation. And after transformation, what you will do? You will do screening of the recombinant colonies that you have observed. So this particular part we have already discussed. Amplification also we have discussed, where we have discussed when the sequence is known, when the sequence is not known. This all we have discussed. So now we are here we are transformation and then screening. So for transformation, so there was a competent cells. So competent cells we have prepared. Suppose we have prepared my competent cells and my competent cells are stored in minus 80 degree. Now today I want to do transformation, so I have taken out one vial. I keep my vial in the ice. Okay, now I have my plasmid. Plasmid means whatever DNA, whatever this thing, ligated product, where the plasmid has your insert, this is your ligated product. So here plasmid means what? The ligated product. Okay, so whatever ligated product I have, now I will incubate my competent cells with that ligated product on ice for 30 minutes. Okay, after 30 minutes, immediately I will give a heat shock for 90 seconds. Okay, not more than that, because it may harm the cells also, it may harm the competent cells. So just for 90 seconds is enough. Then after immediately adding this thing, immediately giving the heat shock, immediately I will add the chilled media for faster recovery of the transformed cells. So if this is my vial, okay, and this is my competent cells, and this is another vial where I have the ligated product. Ligated product. I will add into it 30 minutes incubation. I will put heat shock. Again, I will keep it immediately on ice and I will add the media. And then I will plate. This is my plate. 90 mm dish is there. So someone has, I think, unmuted the mic. So can you please mute yourself, because it is creating some disturbances in the class. If you have any question, you can ask. Otherwise, you can just mute yourself. Okay, fine. So then after addition of the media, we will plate it in the plate, and then we will incubate at 37 degrees for 23 hours. And then we will, after 23 hours, Uh, just a minute. So, Richard, can you please mute yourself? Okay, fine. So, after incubating at 37 degrees Celsius, you will see four colonies. Okay. Now you have to screen these colonies. What is the meaning of screening these colonies? So you can ask that why to screen? If the colonies are forming in the plate, then they must be the colonies which has the product. Otherwise, how will they form? So no, we have to screen because there can be some vectors which are self-ligated. What does it means? It means that during ligation, they have not taken the ligated product. In spite of that, they have ligated again. So those vectors will also give us colony because whatever antibiotic resistance gene is there, that is in the vector, not in the insert. So even if a vector which does not have a ligated product will also give a colony here. So uh, we have to screen for the recombinant clones from the self-ligated vectors also. So therefore, after your transformation comes your screening. So that we will see. So is this general procedure for transformation clear to people? How basic procedure of how transformation is performed? If you have any queries, you can ask me.
Now, can you explain that question 3 once again?
Okay. What is the purpose of giving heat shock during transformation? This question.
Yes.
So
Yes,
basically,
ma'am.
so you have to understand one thing, that you cannot give, first thing you see, that you cannot give stress to your competent cells, okay, because if your competent cells are already in very much stress because you have kept taken out from minus 80 and you are putting from here and there. So if you are giving them stress, so their cell wall will shrink even more, and if they will shrink even more, then there will be even less chances of uptake of the this thing, your ligated product. So you want that your cell wall should expand and it should increase its surface area, so that there are more chances of the ligated product to be taken up. So therefore, when you are giving heat shock, there it is relaxing the cell wall. But you have to understand that that is why it is very important that you cannot say ki, okay, if the heat shock is causing the relaxation of the cell wall, then I will keep it for half an hour so that more and more ligated product or more and more vector it will be taken by it. No. So there is a time that only for some seconds, like for in like I do just for 30 seconds. Okay, so in my lab we do just for 30 seconds. So because after 30 seconds it will start harming your competent cells membrane. So it depends the context of the time up till which you are giving the heat shock. So up to a certain time, like up for few seconds from 30 to 90 seconds, it will help. After that, it will have negative effects. So therefore, when you are giving your heat shock, so it is causing the expansion of the cell wall, and there is more surface area. Your cell wall is getting relaxed. So there are more chances of your ligated product to be taken. First thing. Second thing is that heat shock, because it's a shock. Okay. So so it is. See, you have to understand two things. There are actually I have written it in one sentence, but they are two different. It relaxes the cell wall, and there is the upregulation of factors responsible for DNA recombination and repair. It is different. So because it's a shock, you are giving a shock to the cells. So there will immediately be a mechanism for repair. Like whatever damage is happening because of the shock, there will be a mechanism for repair. So that repair and recombination mechanism, because which is produced because of the heat shock, that recombination mechanism will help in the integration of the the integration of your ligated product with the competent cells. So we are trying to create this to upregulate the recombination and repair pathway by giving heat shock. But this recombination and repair pathway will also be utilized by the insert for its insertion into the competent cells. Okay, it will relax the cell
So
wall.
this recombination and repair pathway is sort of a stress response only, right?
It is a sort of stress response, but see, it depends. You see, here you have to see the balance. Here you have to see कि if you are giving the heat shock for a very long time, so your DNA recombination repair pathway will be upregulated, but your cells will also go under because it is you are giving heat shock for a very long time because you are getting stress. But you have to see what you want. Like you want stress more so that the recombination repair pathway should be upregulated, or you are okay with this much of upregulation of repair recombination and repair pathway, but you do not want a lot of stress. You just want key cells immediately transiently it is expanding, and transiently the recombination repair pathway is sensing that something is happening, it is not normal. Even the expansion of the cell is not a normal expansion of the cell wall is not a normal phenomena of a cell. So the recombination and repair pathway is triggering that, and as soon as the expansion is getting even more detrimental for the cell, you just immediately put it on ice. So that's why whenever you are putting your vial which has your competent cells and insert in your uh, dry bath or water bath, wherever, immediately after 90 seconds, immediately you have to put it on ice. So you have to keep your ice box just besides your dry bath or water bath, so that as soon as your 90 seconds your timer is also there. Even like suppose if I do for 30 seconds, so at I'm very active from 25 seconds. At okay, 25 seconds I just open the lid, I have to immediately keep it on ice, because I know that I cannot give the treatment for a very long time, because although my increased heat shock treatment might upregulate the DNA recombination and repair mechanism even more, but I don't want that much. I have to see that my cells should not go under stress a lot. Is that clear?
Yes, ma'am.
There has to be a balance. Now, the next question is, what is the role of single stranded carrier DNA during transformation in yeast? It blocks the non-specific sites on the cell wall. It makes the plasmid available for uptake. It increases the efficiency of transformation, none of the above, and all of the above. What is the correct answer?
Ma'am, all of the above.
Yes, the answer is all of the above, because the single stranded carrier DNA, it is blocking the non-specific site, and because it is blocking the non-specific sites, it is making the plasmid more available for its uptake, and ultimately because of all this the transformation efficiency is getting increased. Okay, so therefore the answer is E, which is all of the above. Just a minute. Okay, in spheroplast method of transformation, yeast cells are incubated with pectinase, amylase, ribulase. The answer is
Ma'am, practice.
no, pectinase is not the correct answer.
Zymolase.
Yes, zymolase is the correct answer. So the correct answer is option C, zymolase. Next, now we'll come to transformation efficiency. So this example is exactly the same example that you have studied, that sir has discussed in the lecture. So I think someone has also mailed me regarding some of the questions that she was unable to solve in the assignment, whatever is given to you. So I cannot discuss the assignment questions, but of course I can discuss the concepts that are used for your assignment questions. And this transformation efficiency is one of the concept that is used for your assignments. So I think it would be better if we discuss it. So how many of you could understand how to calculate transformation efficiency? Or how many of you have problems in it, or anybody who could understand will try to help the class in solving this question. Transformation efficiency, the question is, you have transformed 1 microliter of 0.01 nanogram per microliter plasmid into 100 microliter of competent cells. You have added 900 microliters of chilled LB media to these cells. From this 1000 microliters, you have taken only 100 microliters, and that you have plated onto the cell. The number of colonies that you got in the colony counter is 450. Calculate the transformation efficiency. How will you calculate it? Anybody who wants to give it a try? Hmm? Nobody wants to give a try for this question? So how many of you have problem in this question then? Okay, one student has a problem. Okay, so that means, okay, I'll then explain the question. So others have understood the question, right? No. Okay, then. So one thing that you need to understand is that whenever we are saying transformation efficiency, you have to keep in mind that suppose this is your plate, okay? And in this plate, after the transformation process, the next day when you come, you see there are some colonies here. Fine. So you have to see colony, the number of colony forming units means the number of colonies that are formed by transforming 1 microgram of plasmid into a given volume of competent cells. Number of colonies per microgram of plasmid. So this is very important, per microgram. Now here it is written that have you gone through that video? There was a video also, I mean the demonstration video. So in that demonstration video, so there was a formula which was given that you can calculate transformation efficiency by number of transformants upon microgram of DNA into final volume of recovery upon volume plated. Okay. Sorry for my bad handwriting. So this is the formula. So transformation efficiency is calculated by the formula number of transformants upon microgram of DNA into final volume of recovery upon volume plated. Okay. So now if you look in our question, so number of transformants. So how many transformants we have got after plating? Number of transformants means that number of colonies that we could observe after counting in colony counter or after in any way. So how many colonies we could observe from the question? How many colonies we could observe? Tell
Around
me.
450.
Ma'am,
450.
450.
280. Where is 280
Around
written?
450.
So in this question, the number of transformants means that number of colonies that are observed. So here the number of colonies observed is 450. Okay, we will take 450. So we are going to do very simply. Abhi, whatever way it was given, anywhere, just leave it. It is the same only, but just it's very simple. Number of transformants is 450. Then microgram of DNA, okay, here it is not microgram, let's take in nanogram. So this much of DNA was plated now, because 1 microliter was only plated and the concentration of DNA is 0.01 nanogram per microliter. This means that in 1 microliter, the amount of DNA which is present is 0.01 nanogram, and they have plated only 1 microliter. So the DNA which is plated would be 0.01 nanograms. Okay. Here we have to convert it into microgram, that we will do later. First thing. Second thing is final volume of recovery. What is the meaning of final volume of recovery? That means that you have taken 100 microliters of competent cells. Okay. But recovery means that because you have given it heat shock, so after heat shock you want to recover so that the cells does not go in very much stress. So for recovery you add media, chilled media. So this much microliter of chilled media you have added, 900 microliters, and 100 microliters of competent cells were present. So the final volume after adding the media for the recovery of the cells is 1000 microliters. So the final volume of recovery would be 1000. So final volume of recovery means that the final volume that you are getting after adding the chilled media. So suppose you have taken 50 microliters of competent cells. Let's say if you took 50 microliters of competent cells and you have added 450 microliters of the media, then your final volume of recovery would be 500. So it depends ki how you have, for final volume of recovery you have to add the microliters of competent cells that you have taken plus the microliters of LB media that you have used for the recovery process. So that the cells does not go in stress. Okay. Upon volume plated, that means that in 100 microliters of competent cells, one is very small, so that's why we are not taking it as 101 because it is close to negligible. So 100 microliters plus 900 microliters, now you have 1000 microliters. But what suppose you, if you want to plate the complete 1000 microliters in this particular plate, then you will not be able to screen individual colonies. Did you understood that why from 1000 they have only taken 100? Because if you plate the, you can say that we should plate the whole number of volume, it would give us more colonies. No, because then there would be so many colonies that it would be like this, something very cluttered, and you have to screen for individual colonies. Then you won't be able to find individual colonies in this case. Therefore, whatever final volume of recovery is there, you take only a few microliters from this. Here they've taken 100 microliters. So I also take 100 microliters only. So the volume plated is the actual volume which is added to the plate. So that is 100. Now calculate this and tell me how much it is coming.
45.
Uh, uh, how much it is coming? Can anybody said I couldn't hear? Sorry. Can you just increase the volume of your mic? Hmm?
45.
Is it coming something like this? Four 450 divided by 0.01, how much it will come? Hmm?
45000 into 10.
Is it clear? 1, 2, 3, 4, 5. Now, this much is the number of colonies which are uh, obtained, but it is not in 1 microgram of plasmid, it is in nanograms. So now for converting into micrograms, what we have to do? We have to multiply by 1000. So this would be 4.5 into 10 to the power 8. And that would be your answer. So what you have to understand is one thing, that suppose if this is given in nanograms, then you have to multiply by 1000. If it is not given in nanograms, if it is given in micrograms, then you need not to do this step. Then you have to just do the formula. Is it clear? Now tell me, where is the problem? Where you could not understand? Tell. Has everybody understood the question, or if you have a problem, then tell me, we'll discuss. Okay, someone has raised the hand. Okay. If someone has a query, you can ask. Anushka. Everybody understood how to calculate
Yes,
transformation
ma'am.
efficiency? Everybody understood how to calculate transformation efficiency?
Ma'am,
Yes.
ma'am, can you explain once more this
Your
question,
voice
ma'am,
is
how to
not
solve
coming
this?
properly. Can you come closer to the mic?
Yes, ma'am. Ma'am, now it is clear.
Uh, a little better. Okay, I can hear.
Ma'am, I'm saying that, ma'am, can you once more explain the question, how to solve these questions?
Okay. So in the question they are saying that you have transformed 1 microliter of 0.01 nanograms per microliter plasmid. That means hmm, that means this is your competent cells and this is your plasmid. Okay. These competent cells are present 100 microliters. In this 100 microliter, you have added 1 microliter of the plasmid, and this plasmid has the concentration of how much? 0.01 nanograms per microliter. Okay. So this is the concentration of the plasmid. So plasmid of this concentration is present with you. From you have added 1 microliter into 100 microliters of plasmid. Is this clear? So now, because the concentration is given to you as 0.01 nanogram per microliter. What does it means? That in 1 microliter, what is the amount of DNA concentration which is present? 0.01 nanogram. Okay, is this fine? Up till here, is it
Yes,
fine?
ma'am.
Okay.
Yes, ma'am.
Now, after adding this much 1 microliter of plasmid, you will incubate it for 30 minutes. After 30 minutes incubation in ice, you will give a heat shock treatment. After heat shock treatment, you will add LB media for the recovery. So this LB media that you have added, it is 900 microliters. Okay. So now this LB media we have added for recovery of the competent cells, so that the competent cells does not go under very much stress. So this LB media that you have added is 900 microliters. So 100 microliters was already present. 900 microliters you have added. That means now total volume is 1000 microliters. So this is the 1000 microliter is actually the volume which is obtained after the recovery. After recovery means after addition of the media. So now you have a vial in which you had 100 microliters of competent cells, 1 microliter of the plasmid, and in this you have added 900 microliters of the LB media. Now your total volume is 1000 microliters. From this 1000 microliter, you have a plate. And from this 1000 microliter, you have taken only 100 microliters, and in drop wise fashion, you add on the plate, and then with the help of a spreader, you spread. And then you keep it, and then overnight when you have kept, and the other day, next day, you observe that there were 450 colonies which were observed. This is the question. Is the question now clear to you?
Yes, ma'am.
Now, the formula is number of transformants upon microgram of DNA. It is very important. Microgram of DNA into final volume of recovery upon volume plated. So the number of transformants, which signifies the number of colonies which are obtained after transformation, is 450 in the question. Microgram of DNA? No, we have nanogram of DNA as of now. We will convert it to micrograms later. So, which is 0.01. Final volume of recovery is 1000 and the volume which is plated is 100. When you will calculate it, you will get 4.5 into 10 to the power 5. But since it is in nanogram and the question says that it has to be in micrograms, matlab the formula says it has to be in micrograms, so we will multiply by 1000. Why? Because 1 microgram is equals to 1000 nanograms. Simple conversion. So your answer will come 4.5 into 10 to the power 8. Now is it clear?
Yes, ma'am, it is clear now.
Okay. Okay, then we'll move to lecture 2. So in lecture 2, we have studied about the DNA delivery in host. So DNA delivery in mammalian cells, the chemical transfection methods, bacteriofection methods, and transduction methods. So now we'll do the questions. The basic principle of liposome mediated transfection method is creating pores through electrical impulse, forming a precipitate through charged DNA, packing DNA into lipid vesicles, and using virus for transfection. So what is the answer?
Ma'am, option C.
Yes. So liposome, from the name itself, it is saying that liposome means something related to lipids. So the answer is option C. Next question is, bacteria mostly used for DNA delivery in plants is Agrobacterium tumefaciens, Salmonella, Shigella, Azospirillum.
And option A.
Option A. Yes, good. Option A is the correct answer. Agrobacterium tumefaciens. Now, plasmid used mostly for plant genetic engineering is RI plasmid, TI plasmid, both A and B, and none of the above.
Ma'am, is it B?
Ma'am, TI plasmid.
Yes. So you have to understand that Agrobacterium tumefaciens has a plasmid, which is this question that we have discussed just now. Agrobacterium tumefaciens. So it has a plasmid which is known as RI plasmid. Agrobacterium rhizogenus is also another strain of Agrobacterium which has RI plasmid. It can also be used, but the mostly used, the widely accepted one is the TI plasmid. Then viral mediated DNA delivery method is called as transfection, lipofection, transduction, bacteriofection.
Ma'am, option C.
Yes, good. Option C is the right answer, which is transduction. So now we'll come to lecture 3. So in lecture 3 was actually, I think, the main conceptual one here, because till now we have studied about transformation. Okay, now we have transformed ourselves. Now we have to see that in this plate we got, suppose how many colonies we got, what are our actual recombinant clones, and what are your non-transformants and what are non-recombinants. We have to observe that. So for that we have to do screening. So this screening can be done by many methods, by blue-white screening, by antibiotic selectivity, the insertional inactivation, which is the same LAGZ gene, antibiotic resistant gene, insertional activation of the CIA repressor, complementation of mutation, screening of then this is all for mostly for your bacteria. For mammalian cells, you can use the reporter gene assays, GFP expression, and after your transformants are selected, then you have to confirm by sequencing, which is Sanger sequencing or Maxim Gilbert sequencing. So we will do the questions also, and I think I will also try to revise just some of the important concepts here because they are very important. And experimentally also, mostly, so in our labs or in most of the labs, mostly what people use is the antibiotic resistance. I'm sorry for that. So mostly we use antibiotic resistance gene. Now the question is, the colorless compound which is a substrate for beta galactosidase in blue white screening is alpha peptide, XGAL, 5 bromo, 4 chloro, 3 indolate, beta galactopyranoside, both B and C.
Ma'am, B.
No. B is
Ma'am,
correct.
option C.
Both are correct. So therefore, the option which is correct is D. So the actual name of XGAL is 5 bromo, 4 chloro, 3 indolate, beta galactopyranoside. Okay. So the option is D. Both B and C. The next question is, if the gene is inserted into the LAGZ gene, what color colonies would be observed?
A.
Blue color, ma'am.
So the answer is A, and we will also discuss this concept later when we will discuss all the questions. Okay, we'll just revise all the concepts. Next question is, a vector contains both ampicillin and tetracycline resistance gene. The gene of interest is inserted into the ampicillin gene. In which order should the cells be plated? Tetracycline, then amp, then tet, grow in some other antibiotic resistant gene, or none of the above?
Ma'am, A.
Sorry?
Option A.
Yes. So can you explain why you are saying option A?
Now, first of all, if we grow in the tetracycline plate, then all the cells um, should grow, and then after when we plate in the ampicillin plate, only those cells which the gene of interest has been inserted will not be able to grow, and then we can check in the tetracycline plate the colonies which are missing in the ampicillin plate.
Correct. So the answer is A, and he has said it very correctly. So there are two plates. Okay, this is your tetracycline ampicillin. Okay, so and this is your vector. Here there is tetracycline resistant gene and there is ampicillin resistant gene. So basically ampicillin, tetracycline, ampicillin, what are these? These are antibiotics. Okay. So they will not antibiotics. So we have any, so suppose we have any bacterial infection. So we take antibiotics. In normal we have cough and cold also we take antibiotics. So why do we take antibiotics? Because it does not allow the growth. So suppose if I am plating my cells in tetracycline plate, so if my gene of interest is inserted here, suppose, so this is a gene which is providing resistance to tetracycline. That means that when I am, as it is if I am taking the vector and I am plating it here, the vector will be able to grow because this vector is resistant to the tetracycline. So tetracycline is not able to kill this. Okay. But if my gene of interest is inserted here only, then this tetracycline resistant is damaged. This gene is damaged now. It is blocked or damaged. So now this vector is not resistant to tetracycline. So therefore the tetracycline plate, it will not grow. This is the basic concept. Now here what I have in the question they have done is that the gene of interest is inserted into the ampicillin gene. So if this is your ampicillin gene, here I have inserted my gene of interest. So I have damaged this ampicillin resistance. That means this plate will not be able to grow in ampicillin plate. Why? Because the ampicillin resistant gene is damaged because I have cloned my gene of interest here. So first I will take my cells and I will plate here and I am getting many colonies here. Okay. Now, from here, again I am plating it here and I got this much colony only. So that means this, just a minute. Hello? Just a minute. So now this colony was not able to grow on ampicillin plate. This colony was not able to grow on ampicillin plate. This means that these are the actual recombinant colonies because they were unable to grow in ampicillin plate. Is that clear?
Yes, ma'am.
Okay. Now we will come to the next question. In the presence of functional CI, what is the appearance of colonies? Lysed and clear, turbid, depending upon the gene, and none of the above.
Ma'am, option A.
No, option A is not the correct answer. It's the wrong answer.
Option B, ma'am.
Yeah, obviously. Yeah. So you have to understand that, okay, I will explain it with it is from lecture 3. Yeah. Okay. Hmm. So virus has two cycles. The first one is the lytic cycle and the second one is the lysogenic cycle. Now whether virus will follow a lytic cycle or lysogenic cycle depends on a repressor which is known as CI repressor. So basically, as the name suggests, it is a repressor. So what it does that it represses the lytic cycle. Okay. So when this CI repressor is active, then the lytic cycle is repressed and the cells undergoes the lysogenic cycle. And in lysogenic cycle, what happens? That because the virus is following a lysogenic cycle, so um, there would be turbidity observed. Why? Because in the lysogeny cycle, the virus is not able to completely kill the host. And because the host is not completely killed, that means the bacteria are not completely killed, then in the plate there would be not clear, clarity would not be there, there would be turbidity. Whereas when your this repressor is disturbed, it is inactivated, then it is not able to repress the lytic cycle, and then the virus will go into the lytic cycle, and when it will go to the lytic cycle, it will lyse the cell. And when the cells are lysed, then in the plate it would be very clear. The plate would be very clear, and there would be plaque formation. So CI repressor is a repressor for the lytic cycle. So the question is saying that in the presence of functional CI, that means the CI repressor is functional. So when the CI repressor is function, then virus will follow a lysogenic cycle and therefore the colonies would be appearing as a turbid colonies. Is it clear?
Yes, ma'am.
Okay. So again the same kind of question. Plaque formation during CI repression suggests functional CI is present, recombinant clones are produced, functional CI is absent, A and C and B and C. What is
Ma'am,
the correct
option E, B, and C.
Yes. So the option is option E. E, B and C. Yeah. Option is this one, B and C. Then yeast auxotrope of uracil means host strain has ample amount of uracil, host strain does not require uracil, host strain does not have uracil, and host strain is not dependent on uracil. What is the meaning?
From option C, it cannot produce uracil on its own.
Hmm. Yes. So the answer is option C. So auxotrop means, so whenever you have, I don't know whether you have heard or not, but there is a term which is known as auxotropic mutants. Auxotropic mutant means that whatever particular gene or whatever we are taking, that is not present in that particular mutant form. So when I am saying that it is a yeast auxotrop of uracil, that means uracil is not present. And suppose I'm taking any other gene, yeast auxotrop of TRP, tryptophan, that means tryptophan is not present. So auxotrof means that auxotrof of a particular gene means that particular gene is absent in the host strain. So the answer is C. Next question is, in complementation of mutation of yeast, positive and negative selection means what? Recombinant clone has the functional gene, and so this means this is the positive selection, this is the negative selection. So positive selection means recombinant clone has the functional gene, and negative selection means recombinant has non-functional gene. B, recombinant clone has the non-functional gene, it has functional gene. C, recombinant convert into cytotoxic agent, recombinants have no cytotoxicity. And D, both A and C. So what is the correct answer?
From option A,
Okay. Anybody else who wants to say anything? Who wants to give a try? Any other option? Do you agree
Option
with this
D.
answer? Or so A. So we have two votes for A. Okay. It is the correct, but we'll just see with the help of, here we'll see, and then we'll just uh, quickly discuss this concept, and then we'll move further. So basically, when we talk about complementation of mutation, it means that whatever mutant host strain they are taking, that is an auxotrophic strain. That means it does not have a particular gene. Okay. So here what it is written, that in this approach, a mutant host strain can be used to screen the plasmid containing the missing gene, and the transformed will grow only if the gene product from the clone will complement the function. So what does it mean? Ki this is the host strain, and suppose I am taking it is auxotropic for uracil. So this host strain is devoid of uracil, or the uracil is inactive. Now, because for its synthesis, it requires the uracil biosynthesis pathway. So this uracil biosynthesis pathway will only be completed when the vector, the recombinant that you have produced, that has a functional uracil. If it has a functional uracil, then the host strain will take the functional uracil from the vector, and then the pathway will be completed. So this is a basic definition, but when we study about complementation, it can be of two types. First is a positive selection, and second one is a negative selection. So a positive selection means that suppose this is a host, and host does not have uracil. Uracil is absent in host, and this is a recombinant clone. It has the uracil. Recombinant clone has the uracil. So when it is transforming, so the biosynthesis pathway is getting completed because uracil is present, and therefore whatever colonies that you are getting, uh, that are going that are cultivating, because uracil is getting from the recombinant clone. So why it is called as a positive selection? Because the clones that are growing because uracil is present in the recombinant clone. Okay, so that means the functional gene is present in the vector. So when a functional gene is present in the vector, then that is known as a positive selection. And negative selection, what happens is that suppose there is a gene which is present in the vector, which is not good for the I mean, not good, but if something externally, if you are providing an agent which can hamper the growth of the host strain. So what is written that in the negative selection, a chemical compound is added to the media which will be converted to a cytotoxic agent in the presence of the gene product, and as a result, it does not allow the growth of the wild type strains. But host strain transformed with the recombinant clone has non-functional gene product and grows in the presence of the compound in the media. That means here the host strain was able to grow. Why? Because of the functional gene which is present in the recombinant clone, which is helping in its growth. Here the host gene is able to grow because the recombinant clone has a non-functional gene. Therefore, the cytotoxic agent is not getting produced, and therefore the host is able to grow. Host strain is able to grow. So, in both the cases, the recombinant clones are helping the host strain to grow. But here it is helping by positively activating the biosynthetic pathway. Therefore, it is a positive selection. Here it is helping by negatively regulating, by not providing that cytotoxic agent which can hamper the growth. Therefore, it is a negative selection. So, in negative selection, your wild type is not able to grow. So wild type will not grow because of the presence of the cytotoxic agent. But your recombinant does not have that functional gene. Therefore, it is able to grow. So in positive selection, you have a functional gene, and in negative selection, you have a non-functional gene. So if we look into the question, positive and negative selection means, so in positive selection, there has to be a functional gene because functional gene will complete the biosynthetic pathway. Whereas in negative selection, the recombinant will have a non-functional gene so that that cytotoxic agent is not formed. Cytotoxic product is not formed. So the correct answer is A. Now we come to the next question. In complementation of mutation of yeast, negative selection will produce functional wild type, functional recombinant clones. Both wild type and recombinant would be functional. No colonies are functional. Replica pait is needed. What is the answer? Or the question is not complete? Tell.
Now, functional recombinant colonies.
Yeah, so actually, the question is not completely mm, elaborated because it depends. So here the question, it is written in complementation of use, negative selection will produce what? So negative selection will produce, um, it depends on the context that what we want to say, that this negative selection will produce functional wild type colonies if recombinant clones are not present, and it will produce functional recombinant clones if the gene of interest is non-functional, that is inserted into the host strain. So uh, mostly it should be this, but then some might get confused with functional wild type colonies also. But you have to be very, like, you have to choose the best possible option, because questions like these might confuse you, because here the in the question it is just written negative selection, they are not completely elaborated in what context. But anyways, the best option here is functional recombinant colonies.
Ma'am, the wild type would like never be able to function because they produce this toxic substance and all that, right? So, I mean, how can we obtain a functional wild type?
You are correct. So in negative selection, good point that you have raised, that in negative selection, sorry, I think I got confused with this. Yeah, so he's saying correctly that in negative selection, wild type colonies will never be functional because in wild type colony that cytotoxic gene is present. So the answer would be functional recombinant colonies also. Correct. Good. Now, the next question is, what is the substrate for luciferase enzyme? Oxyluciferin, D-luciferin, luciferin aldenylate. Just a minute. So what is the answer?
Option B.
Option B is the answer. Yes. So luciferase, the substrate of luciferase enzyme is D-luciferin. Luciferin, and what is the ultimate product that is produced?
Ma'am, oxyluciferin.
Yes. Oxyluciferin is the product which is produced, and the substrate for luciferase enzyme is D-luciferin. Next question is, plasmids are used as cloning vector. Why? What is the basic property which helps the plasmid to be used as cloning vectors?
Ma'am, option B.
Self-replication in bacterial cells. Yes. So option B is the correct answer. So plasmids can be used as cloning vectors because they have the ability to replicate inside the bacterial cells, because any cell for that matter will multiply in culture when you are providing it all the nutrients. But important is that plasmid has to be inserted into the bacterial cell, in bacterial cell whether it is able to replicate or not, because there you have to, when you are putting it overnight or whatever, you cannot supply all the time external nutrients. So there, if it has a property of self replication, it is easier for us to manipulate it. So the basic property why plasmids can be used as vectors, because they have the property of self-replication because of the origin of replication gene which is present in the plasmids. And now that we have so much studied about recombinant, the scientists have so much studied, it is now a very well elaborated field. Now we can manipulate, like we can artificially create the kind of plasmids that we want by cloning the kind of genes that we want to insert in the plasmid. So suppose if this is a plasmid and I want an origin of replication of a strain X, origin of replication of plant, origin of application of bacteria, origin of replication of a of a mammalian. You can do both of them, and you can make a kind of shuttle vectors also. So depending upon the kind of origin of replication it is present, it will replicate in that host strain. But the basic concept remains the same, that plasmids can be used as cloning vector because they have the ability of self-replication. Then blue-white screening is used for screen the recombinant vectors, detect gene mutation, identify desired chromosomal DNA insert in plasmid vectors, detect host DNA.
Ma'am, option A, ma'am, screening recombinant vectors.
Okay, yes. And anybody else who wants to give a try?
Ma'am, option C.
Option C. Okay. Anybody else who wants to give a try? So,
Ma'am,
option
option
C,
A.
you have said option C. Can you explain why you chose option C? Identify desired chromosomal DNA insert in plasmid vectors. Can you explain your answer? Why you chose option C? Narayan has a query. So yeah, you can ask. Okay. Please explain blue-white screening and what plasmids we use and cannot be used. What plasmids can be used and cannot be used? Please provide examples. So whatever plasmids can be used and cannot be used, it depends on the context of what you want to do. So suppose if I want to clone a mammalian gene, I cannot use a PBR322 or a PUC19 for that matter, because because they are not able to replicate in mammalian cells, because they don't have that mammalian origin of replication. So what plasmid you want to do depends on what is your question, what you want. Okay. So examples, it depends on the context. And blue white screening, okay, I will explain in the end what blue white screening is. Anyways, so for the question number 22, what should be the answer? So one girl has said option C. So can you explain why you chose option number C? Hmm? You said the correct answer only. Just explain the point. Uh, okay, so the correct answer is option C. This we will discuss when we will do the blue white screening at the end. Anyways, basically they. Okay, I will explain in the end only. That would be easier for you. Question number 23, generation of recombinants by random cloning of. So have you studied about shotgun cloning? Have you studied up till now? I don't think so. Was it there in shotgun cloning was there? Anybody?
Not in the videos, ma'am.
It was not in the videos, right? So we'll just leave the question. So basically, the answer is shotgun cloning. So generation of recombinants by randomly cloning fragment of total DNA from an organism is called as shotgun cloning. So I'll just leave the question. Let's see whether in the subsequent lectures, sir will explain shotgun cloning or not. If not, then we'll discuss this cloning. So just remind me if this is not completed or covered in the subsequent week class. Okay. So
Ma'am, for this, can't the genomic library be the answer? I mean, we are creating separate recombinants and
sorry, I couldn't get your question.
ma'am, for this question, can't genomic library be the answer?
No, genomic library cannot be the answer. So shotgun sequencing, shotgun cloning is a different kind of cloning. So we will discuss it. I just want to see whether sir is covering this topic or not. Genomic library is different. Genomic library means genomic library is the library of the library of the genome of a particular organism. So for making that library, we clone that fragment, but that cloning is different from a genetically modified organism cloning. So this cloning we are doing because that library is very long. Okay, we have sorry, the genome is very huge. So it's better to cut the genome into smaller fragments and then clone individual fragments and then you kind of align them. Okay. So, but if you are want to generate recombinant, then shotgun cloning would be a better approach, although there is a very slight difference between how to create genomic library and shotgun sequencing. But there is kind of overlapping that happens when you do shotgun cloning that I don't want to discuss it just right now. Let's see whether sir is explaining in the subsequent classes or not. Otherwise, we will discuss this point. Anybody please remember and remind me if this is not discussed. Yeah, okay, just a minute. What are PET series plasmid? Are they recombinant or not? So Narayan, so what do you understand about recombinant and non-recombinant plasmid? When a plasmid will become recombinant, tell me. Any series of plasmid for that matter, whether it is a PET series, whether it is a puck series, whether it is a whatever series it is. When a plasmid will become recombinant? Yes. So when we add external genes. So how can you say that PET series plasmids are recombinant or not? So any series of plasmid will become recombinant only when we add a particular external gene into it. Otherwise, they will just remain as in their own original form. So whatever plasmid are there, so these plasmids we will take and when we will add the our gene of interest into it. So is PET series covered in your syllabus? Was PET series there in your syllabus? I don't think so. So like in the class also, sir has mentioned that there are n number of plasmids. Okay, so and it is not basically uh, we cannot discuss all the plasmids. So I will restrict my discussion to the topics that were discussed by the sir. So PET is a completely different series of plasmid where the expression depends upon T7 RNA polymerase, and it would be very difficult for me to cover this in a 2-hour class. So like if you have any queries, I can make a separate video, not now, after this 3 months is completed. But as of now, I cannot cover the point because it is beyond the scope of the class to cover each and every different kind of plasmid. As of now, what you can understand about the PET plasmid is that whatever gene of interest that you are cloning into the PET vector, so it is in the control of T7 RNA polymerase. Okay. See, how you can say that is a recombinant or non-recombinant? Any plasmid will become recombinant only when you are adding a gene of interest. No, it's a basic concept, Narayan. So, how can you say that whether this plasmid is a recombinant plasmid or not? Obviously, when you will add some external gene, then it will become a recombinant plasmid. If I am purchasing a plasmid from a company at gene, then it will not come into recombinant form. It will come in its original form. I will make it recombinant by adding my external gene to it. Okay. So any plasmid will be recombinant only when we add any external gene to it. Yeah, so I'm telling you the basics that the PET plasmid is a plasmid which is in the control of a bacteriophage system, that is T7 RNA polymerase. So T7 RNA polymerase is there in PET plasmid, and whatever transcription and translation is happening in PET vector, that is under the translation machinery of T7. Okay. And basically, there are two kinds of plasmid. The one is the cloning plasmids, and other one plasmid is used for expression plasmid. So cloning plasmids are the plasmids which are just used for cloning. You do not want to express your gene of interest. Whereas expression plasmids are the plasmids where you're not only cloning your gene of interest, but you also want to express it. So when you want to express the gene of interest, then you require transcriptional and translation machinery as well. So transcription and translation machinery can be provided when you have that regulatory system. So PET is that kind of a plasmid which can be used as an expression plasmid because it has a T7 RNA polymerase gene. Okay, this much only we can cover as of now for this PET plasmid. Okay. So the next question is, in lac operon, gene product of lac Z gene is?
Beta
Ma'am, beta
galactosidase.
galactose.
Yes, correct. Answer is A, beta galactosidase. Next question is, in Griffith's experiment, the pneumococci strain produced isolated from dead mice was live smooth, dead rough, live rough, dead smooth cells. What is the correct answer?
Ma'am, B.
Okay, ma'am.
No, option B.
Ma'am, it's A.
Yes, the answer is A, live smooth cells. Then DNA polymerase uses dash as a substrate. What is the answer?
A nucleotide.
Yes. So what a basic question. What is the difference between a nucleotide and nucleoside? Very basic.
A phosphate group when
Phosphate
attached
group.
to a nucleoside is called nucleotide.
Yes. So next question is, guanine specific cleavage in Maxim Gilbert method is done by using formic acid, hydrazine, DMSO, pyridine. Guanine specific cleavage. Tell me, think. You can go through the slides to answer.
Ma'am, C.
Yes, the correct answer is C, DMSO. Main enzyme component of Sanger sequencing is?
Polymerase.
Yes, polymerase is the correct answer. Next question is, sequence of structural gene in lac operon is? Tell.
Ma'am, option D.
Ma'am, option A.
Option A is not correct.
Ma'am, option
Ma'am,
D.
C, ma'am.
No. It is option D, ZYA. Next is, lac operon will be turned on when? See the options and tell the answer.
Ma'am, option C.
Option C, lactose is more than glucose. Yes. So option C is the correct answer. Lactose is more than glucose. So now we have finished the questions now as we do in our every class. So now we'll take up the topics that you could not understand in the last week lectures. So now you tell me that what topics you want me to explain. Any topic that you could not understand from the previous week lecture that we'll take up. Blue white screening. Okay, so first we'll start with blue white screening. It should be in the, yeah. Hmm. This is blue white screening. This is the basic in it is in lecture 3. Okay, so in lecture 3 this is the general principle of how the blue white screening is, the blue white screening is, and then how blue white screening is used for screening is your insertional inactivation. Okay, so basically we know that we all have studied about an operon which is known as lac operon. Okay, so lac operon, the three genes which are very important are Z, Y, A. So this Z, lag Z gene, which is present in the lac operon, it is responsible for the synthesis of enzyme which is known as beta galactosidase. Okay. Now this beta galactosidase is required for the proper functioning of the lac operon. That all is clear. Now in blue white screening, what is done is that this beta galactosidase, it needs a product, a substrate. So that substrate is known as X-gal. So this is a synthetic substrate that is used in blue white screening. So what happens is that this X-gal is a colorless substrate, and when beta galactosidase it acts on X-gal, it produces a colored product, and that colored product is of blue color. Okay, so when functional lag Z gene is present, then this beta galactosidase will act on X-gal, and then this will produce a product which is blue in color. Is this clear? Now I'll just read it out that the most popular system to exploit this feature is blue-white screening, where a colorless substrate is processed to produce a colorless product. So we have studied that lac operon has three genes, Z, Y, and A. Now ZY produces an enzyme which is known as beta galactosidase. This beta galactosidase will act upon X-gal. This X-gal we will provide, and this X-gal will be used by beta galactosidase, and then a product is formed which is blue in color. A colored product is formed. Now this compound is a substrate for beta galactosidase. Now the beta galactosidase is the product of LAC-Z gene that we have already studied of the LAC operon. So what we do is that in whatever host strain we are using, here the LAC-Z gene is non-functional. How it is non-functional? Because the N-terminal region of the LAC-Z gene is not present there. Okay, so here it is written, the host contains LAC-Z gene without the initial region, whereas vector contains alpha peptide to complement the defect to form the active enzyme. So that means that how you will do the blue-white staining is that we understood that LAC-Z gene will produce beta-galactosidase, and this beta-galactosidase will act on X-gal, and therefore a colored product which is of blue color will be produced. So now what we do is that there is a host strain which does not has a functional beta-LAC-Z gene. So if the host does not have a functional LAC-Z gene, that means functional beta-galactosidase will not be produced. That means X-gal will not be utilized, and blue-colored substrate would not be blue-colored product is not formed. So what we do is that we make a vector which contains the N-terminal lesion which is missing in the host. So the host has a LAC-Z gene in which the N-terminal region is not present, but the vector that we are providing that has that N-terminal region. So when we insert the vector, when we plate the vector into the host cell, so the host will take the N-terminal region from the vector, and therefore a functional LAC-Z gene will be produced. And hence, when you will provide X-gal externally, then a blue colored product will be formed. Okay. So basically, the N-terminal region from the vector will join or reconstitute with the C-terminal region of the LAC-Z gene which is present in the host. And therefore, a functional LAC-Z gene would be produced only when you supply the vector. And when the functional gene is produced, the blue colored product is formed. Okay? Is this much clear? Is this clear to you? I just have to check it here. Now, we know that this is my plate host host strain. This is my host strain. Okay, this is the LAC-Z gene. This is the N-terminal. This is the C-terminal region. My host strain has only the C-terminal region. The plasmid that I am providing, it has the N-terminal region. Now this N-terminal and C-terminal region will join, and therefore a functional LAC-Z gene is produced, and therefore X-gal, when we are supplying externally X-gal, it would be utilized by the beta-galactosidase, and then blue colored product would be formed. But what will happen when I am cloning my gene of interest in the LAC-Z gene, in the N-terminal of the LAC-Z gene? Then what will happen? Will there be a formation of a functional gene then? No. See, this is the LAC-Z gene, functional beta-galactosidase. When we provide externally X-gal, blue colored is formed. But when we insert our gene of interest in the N-terminal region of the LAC-Z gene which is present in our insert, then this LAC-Z gene is disrupted. And because this LAC-Z gene is disrupted now, this will not be able to produce a functional LAC-Z gene, and therefore beta-galactosidase will not be functional, and therefore colorless colonies would be formed. So here the term is saying insertional inactivation of LAC-Z gene. That means the insertion of the gene of interest causes the inactivation of the LAC-Z gene, and hence because the LAC-Z gene is inactivated, therefore functional beta-galactosidase will not be formed. Therefore, X-gal will not be utilized for the conversion of a blue-colored product, and therefore you will get colorless colonies. So when your gene of interest is present, you will get colorless colonies. When your gene of interest is not present, you will get blue colonies. Is this clear? Is it clear or not? Or if you have any doubt, you can ask me. Okay. So That means this blue white screening basically depends upon insertional inactivation. So blue white screening is nothing but lag Z. In one case, if the gene of interest is not there, then lag Z is functional. Therefore, this N-terminal region of the lag Z will combine with the C-terminal region of the host, and therefore a proper functional whole lag Z gene is produced, and therefore they will form blue colonies. Whereas when you are cloning your gene of interest in lag Z only, so when you are putting your gene of interest here in lag Z, so you are disrupting the gene, you're disrupting the lag Z gene. And because the lag Z gene is disrupted, so no functional beta galactosidase, no functional beta galactosidase means no blue color, that means colorless colonies would be formed. Okay. Any other next question, next any topic that you could not understand? Anybody else?
Ma'am, can you explain the Maxim Gilbert method?
Okay. Maxim Gilbert, it was in the last. So basically, for sequencing, there are two approaches. Okay, the first approach is your uh, um, enzymatic enzymatic approach is there, or dideoxy chain terminal approach is there, and the other one is a chemical approach is there. So your Maxim Gilbert is basically a chemical approach for sequencing. So here what we are doing is that whatever sequence of DNA we have, so first with the use of chemicals, we are modifying the DNA. Okay, so ATGC, whatever ATGC we have, so with the help of certain chemicals, certain respective chemicals, we are modifying the nucleotides and subsequently we are cleaving it and then we are doing the this thing sequencing. So what is written that in this method a 3' or 5' radio labeled DNA is treated with base specific chemicals which randomly cleaves the DNA at their specific target molecules. These fragments are then analyzed on a high resolution polyacrylamide gel and then the radiogram is developed. So what is happening? This is my DNA. Sorry. I'm so sorry. Suppose this is my DNA. And this is the 5' end and I have radio labeled it. In reaction 1, what I am doing is that I am adding dimethyl sulfate. Now this dimethyl sulfate will modify G. Okay. So if it, I think there should be some diagram also here. Okay, that will come later. So basically, so there are 4 different kind of chemicals which will modify the base and it will cleave there. So suppose if I am giving dimethyl sulfate, so dimethyl sulfate will modify G and there would be a cleavage at G. When I am giving formic acid, so it is G plus A, that means it is modifying
Ma'am, what does this piperin do exactly? Ma'am, it will be C. C. Ma'am, C.
Like platy E cells or other 293T cells are there. So basically what I do is that first I produce activating that gene. Insertion of the insert is inactivating that gene. Insertion of the insert in lag gene will inactivate lag Z. Insertion